Hello, everyone. Thank you, Gavin, for the introduction. So my background is slightly different. I'm an ecologist, and what I'm interested in is actually the environment. And my main area of research is looking into the pathogens currently in the environment, and also modeling the spread and emergence of infectious diseases in wildlife. Today, I'll be talking about using genomic surveillance for pathogens and antimicrobial resistant detecting in the environment using PacBio's new sequencing platform, Onso. But let me first introduce our lab. So we are the Wastewater Epidemiology Lab. We were formed at the start of the COVID-19 pandemic, and we carried out the SARS-CoV-2 wastewater surveillance program in Northern Ireland. What we were doing is we were basically detecting presence, absence, and concentration of SARS-CoV-2 in wastewater across the whole country. And we were also doing a whole genome sequencing of the virus in order to track any new mutations or any new variants arriving into the country. We were actually the first one to detect the arriving of Omicron in Northern Ireland. But we work with a lot of other viruses. For example, in 2022, there was an outbreak of acute hepatitis of no etiology in children. World Health Organization recorded over 1,000 cases across the globe. And a lot of those kids actually tested positive for adenovirus, specifically serotype F41. So in this case, we decided to implement a wastewater-based epidemiology approach and look into the concentrations of F41 in wastewater before and during the outbreak. And what we find out is that actually the concentration of F41 in wastewater increased with increase of clinical cases. And one step further, we also did whole genome sequencing using a tiled amplicon approach. So the primers were developed by the Arctic Network, who also developed the tiled primers for SARS-CoV-2. And we found out that actually the sequencing in wastewater were homologous to the one in the clinical cases. We've done some work on RSV and also on influenza, and we are about to publish a paper at the sequencing method using nanopore for whole genome sequencing on influenza, both human and avian. We actually managed to detect avian influenza in wastewater, and we're doing some work in looking into avian influenza in the environment as well. We are also part of PathSafe. This is a pathogen surveillance in agriculture, food, and environment program. It's a UK-wide program, and the main goal is to detect and track foodborne diseases and antimicrobial resistance. And our lab specifically focuses on care homes. In care homes, we have a very high rate of antimicrobials prescription, and even higher in Northern Ireland in comparing to the rest of the UK. And also, as we probably all can assume, the residents at those care homes are very susceptible to different infections. So we take environmental samples, we take wastewater, we take air samples, and we also take swabs, surface swabs, and we apply both methods, culture and molecular. So for culture, we do pure cultures, identify them using mass spec, and from there we can look into the minimum inhibitor concentration of different drugs. And for the molecular methods, we use qPCR for targeted approach, looking at specific genes of interest, of antimicrobial resistant genes of interest. And then we also currently use nanopore for doing, and Illumina as well, for doing shotgun metagenomics as well as whole genome sequencing of pure isolates. Quick shameless plug. So Biosphere is a spin-off company that is within our lab, and I'm also part of it. And we're providing kind of commercial services for molecular and culture work, specifically focused around pathogens and AMR. So as you can see, our lab is quite used to work with a variety of samples. So on one hand, we have wastewater samples, which are very high biomass, but they're full of PCR inhibitors. On the other spectrum, we have, for example, air samples, which are extremely low biomass. On top of this, our targets are often at very low concentrations. For example, looking for bRNA viruses in honey is literally looking for a needle in a haystack. So this is one of the reasons why last year we decided to purchase Onso. As Gavin mentioned, I think we are the first commercial customers globally. And what was the most attractive part for us was the high quality. And we are talking about quality of 40, 1 in 10,000 error rate. On top of that, we have a lower detection limit. And also, it means that we can sequence our samples at lower depth without actually compromising on the quality of our results. This is a slide I stole from PacBio very quickly for people who are not part of Sarah's workshops, looking at the difference between sequencing by synthesis and sequencing by binding. The main difference is that the binding and incorporation steps are separated in the sequencing by binding methods. You have, and it's thanks to this reversible terminator that you add it to the prime end of the chain, and it doesn't allow elongation of your chain, and then you can add your base, interrogate, take the photo, and then it's removed, and then the chain is extended very briefly. So we decided to, here, very quick disclaimer, we've had Onso for only several months, so we haven't done any kind of extensive validations. We haven't managed to compare it to our sequencing platforms, NanoPo or Illumina. However, I'm going to talk about our experience so far. So we decided to test Onso with a lot of different samples. Our main goal was to make PacBio's team life extremely difficult. So we decided to use wastewater and air samples, which are ours from nursing home, but also samples from other research groups across the university. So last year, last summer, there was a huge algal bloom in Loch Ness. Loch Ness is the biggest freshwater lake in Northern Ireland. Oh, not only Northern Ireland, in the whole UK and Ireland. It was an ecological disaster. So we managed to collect surface water samples as well as algal mud samples along the shore. A research group in our university is working on population genomics of the damselfly. It's an endangered species. So it's quite difficult to work with as you use only leg. You have a leg from the damselfly. They're endangered, so that's basically all you can get. And actually extracting DNA from insects it's very hard. And then we had some clinical samples. Very briefly about our clinical samples. I include this slide because I imagine there'll be a lot of people interested in cancer. I know nothing about cancer. So this is a research group at the Experimental Medicine, the Center of Experimental Medicine, Queen's University, and they're trying to work at kind of novel method to diagnose cancers with their poor outcome. And what they're looking at is the circulating tumor DNA in the blood. Okay. I'm going to walk you through all the wet lab procedure. We got a lot of different samples, and clearly we need to kind of have a slightly different approach to them. We had the high biomass samples, which is genomic DNA, which is wastewater and algal mud, so we need to do some fragmentation. Then low biomass genomic, we needed also, apart from the fragmentation, we needed also to incorporate a PCR step, so it's a random amplification step, in order to get sufficient amount. So we need around 200 nanograms of DNA to do these runs, and a lot of people talked about micrograms here. We don't work with this. 200 nanograms, even for us, is really a lot. And then we had low biomass, and this is the cancer samples, which are short fragmented, so we have to do is just do a PCR step. And then the rest is kind of a typical library prep steps. We got N-repair, A-tailing, adapt editing, and cleanup. Not all of our samples behave as we wanted them. For example, for wastewater, the fragmentation probably was not sufficient within the library prep kit because we still had big fragments left. So we are thinking in the future to probably do or play around with, it's an enzymatic fragmentation, with the amount of enzymes we add over duration, or maybe do mechanical or some other prep processing step. Our air samples, again, very, very low biomass. So for the PCR was not enough. So we didn't, we use an external kit, and we use those kind of long isothermic amplification steps in order to generate enough material. And then the next step is library sizing and quantification, which for me it was the most labor-intense step and the most probably time-consuming. We did tape station, which is kind of a standard in our lab, but we needed to do a PCR amplification in order to generate sufficient amount of DNA to be actually able to visualize it, and also we use qPCR to do the quantification. The good news is that PacBio does provide the qPCR assays, so we're targeting the adaptive sequencing, so you have to do it before pulling out together. And clearly, if you have 96 samples, random triplicates plus your standards. It's for at least 4 plates that you need to qPCR. And then as well, PacBio provides a lot of different kind of resources in order how to quantify and actually being able to put your samples together. Cluster generating and sequencing. So until actually the PacBio, the Onso arrived in our lab, we didn't realize it's going to be 2 separate equipments. For some reason, we thought it's going to be just one. And we even didn't think about enough space for the 2 of them. So one is your cluster generator, and then the other one is the sequencer. So you put your float cell in the cluster generator. It takes around 5 hours for the cluster to generate, and you actually then have to physically take the float cell and move it into the Onso sequencing, which is quite different comparing to all the other sequencing platforms we've worked so far. Also, the interesting thing about the float cell is actually it has 2 lanes. So in our case, we loaded 2 libraries. One was the low biomass, and the other one was the high biomass. But you can also load your library in different concentrations in order to kind of validate and look into your cluster density generation. And now coming into results, I'm not going to talk about the significant kind of biological significance in our results, but more about kind of the quality, the type of data we got, and also the different bioinformatic tools that we use on the data. So I'm just going to give you a lot of different files. I'm going to generate a lot of them. Half of them I have no idea what they are. A lot of them are helping you with kind of giving you information regarding the matrix of the run itself and how it went. You got also all your raw photos and the most important FASTQ files. I forgot to mention that we did 300 cycles paired end sequencing. So on the bottom in the table you can see expected versus our results. You can see our cluster density was a little bit lower than what we'd expect, and as well the number of reads per lane. So we'd expect over 400 million reads per each lane, and remember there are 2 lanes in the float cells, and we got on average around 356. Again, probably as a result of pulling the libraries and playing around with concentration and actually of individual samples and the loading concentration. And right, there's a popsicle graph. So the samples, the kind of lower half of it is our wastewater samples. So they generated way much more rates comparing to the other samples. So that's something else that we need to take into account, that different samples behave differently. And we did pull a lot of variety of samples into one run. We'll usually probably separate them. Yeah, so this is, I guess, the most exciting result or slide of my presentation. I guess most of you are familiar with the quality score graphs. So on the X, we had the position of a cycle, and then on the Y is our mean quality value. So we got quality quite over 40, and for some of the samples of the wastewater, we can see that quality is over 50. And we actually get the same result across all our samples. The damselfly at the bottom, you see there is at the end. It doesn't look very good, but we think it's some kind of a contamination throughout the extraction process because we had non-insect DNA that was in high repeat, and it was very high GC content. Lucky us. Next, looking into the bioinformatic pipelines that we're using. So this is just quick looking into the microbial biodiversity. This is in the air from nursing homes in a couple of different locations. So we had like a kitchen, the foyer, which is the entrance to the nursing home, and then a common room where like all residents gather. We usually use Bowtie2 to remove any kind of human DNA, because you will have a lot of human DNA in there. Then classical Kraken2 for classification, and then bracking for abundance estimation. We didn't have to do any kind of a quality filtering steps because the quality of the data was so high. We also done some metagenomic assemblies. So this is from the Loch Ness samples from the surface water and the mat. We use metaspades for our assemblies. It is more computational intense and requires more time in comparing to other kind of assemblies, but what we find so far is that we get the best results. I think initially we allocated only 50 gigabytes of RAM, which was clearly not enough. And we had to restart. The water samples, you see they have higher rates in comparing to the mats are still being processed, they're still running. Yeah, for some of them it's been 28 days. Overall, as I mentioned, without doing any extensive validation, so far the Onso result seems quite promising. I'll be honest, I was quite skeptical. I did not expect this high quality data, especially from some of our wastewater raw sequencing data. We are yet to compare the results to, a lot of the samples have been run on Nanopore P2 as well as Nova6, so we are yet to compare the results. And we also like to minimize assays in our lab, so we are yet to see how a minimized library prep version will work on Onso. I just want to thank everyone in my lab, so John, Dedre, Andrew, Hannah, and Martin, who are part of a wastewater sequencing lab of other research groups across the Queen's University, our funders, and of course the PacBio team. Specifically, well, Sarah, Nick, Sam, and Gavin, who've been incredible, and also all the technical team who've come and installed PacOnso. We're very open for collaborations, so my email, and if people still use Twitter, you can always find us. And yeah, thank you. And thank you for staying awake.